Okay, good morning and welcome back into the TYBSc Zoology Mammalian Histology class. We have syllabus point 3.5 that gland associated with the digestive system. In that we will see today a pancreas as an endocrine and exocrine gland. Pancreas, both way it works as a exocrine gland as well as endocrine gland. So today we'll see detail about that pancreas histology. Here in front of you there is a diagram showing the pancreas anatomy, the exact location of pancreas in a, here it is related with the human pancreas. So as a representative of a mammalian pancreas, so they are located in the C-shaped curve of a duodenum just below to the stomach, and in the curve of duodenum they are located pancreas, that all substance of a pancreatic tissues are spread and they form a elongated body in a C-shaped curve of a duodenum. From this pancreas there arises a ducts, a tubes that is called as a duct of Santorini and duct of Wilson. Duct of Santorini and duct of Wilson. This duct of Santorini fuse with the bile duct and open into the duodenum, whereas duct of Wilson separately open into the duodenum. But we are not supposed to go in a much detail about the anatomical position, so we'll fastly cover this portion. Here, the pancreatic anatomy, the glands with both exocrine and endocrine function. Already I told they serve as a exocrine as well as endocrine functions. The size in a human, it is about 15 to 25 centimeter long and weight about 60 to 100 gram. Location is a retroperitoneum. That is, they located behind the peritoneal membrane, and the second lumbar vertebral level it is located extended in an oblique transverse fashions, and parts of pancreas are head, neck, body and tail. Now here again you can see how these ducts open into the duodenum, that called as a minor duodenal papilla and major duodenal papilla. This is all pancreas portion, yellow color pancreas portions here, and these are the duct, principal pancreatic ducts is there. It collect all the pancreatic juice and pour it into the duodenum. Pancreatic secretions, if we consider the exocrine secretion of pancreas, then they produce these protease which are endopeptidase and exopeptidase. Here, with these precursors of enterokinase, the trypsinogen converts into trypsin and they activate all other precursors. Then there are some nucleases secreted by the pancreas as a DNase and RNase, enzymes, and pancreatic amylase, hydrolase, starch, and glycogens, and pancreatic lipase as triglycerides broken into the fatty acids and glycerol. Then regulation of this secretion take place with the help of this secretin and pancreozymes and gastrines and autonomic innervations, vagus nerves. Okay, we don't have to go in detail of this physiology. We are much more confined to the pancreatic histology here. So pancreas gland that has it's a partly exocrine endocrine. So main bulk of a gland being constituted by the exocrine parts, and in between these exocrine parts there are scattered some endocrine area that is called as the isolate of Langerhans. This isolate of Langerhans word is related with the endocrine functions. Now, these exocrine pancreas secrete enzymes that plays very important role in the digestion of carbohydrate, protein, and fats, as we already seen that this pancreas they they are, you can say, this form of a serous gland or compound tubulo-alveolar gland, means they form a alveoli, a group of cells are formed with interseparating septum, and so it is called as a alveolar gland. That all group of basic pancreatic cells are separated into a vacuoles, and that vacuoles or alveoli, they together then club and they form that compound tubular alveolar gland. A delicate capsule surrounds this pancreas. That septa extends from the capsule into the gland and divide into the lobules. Okay. In between somewhere there lies scattered this islet of Langerhans. Now, in this color photograph, when you take a section of the pancreatic portions, now here you see all these pinkish color structures with these blackish color dots present in that pink color structure. This is how we correlate that blackish spots are nucleus and this pinkish color is a cytoplasm. So if you see the magnified view of each this small pinkish color structure here, you'll find they are the acini. Acini means a cuboidal cell lining and pointed centrally a hollow lumen. Acini is a structure in histology where from all the sides there is a cuboidal cells are present and the central lumen is open. But here, if that acini is of a pancreas, how to identify that? You will find that nucleus is at the periphery. So all the nucleus are arranged to the periphery of this acini, and that is the one characteristics of identification of these pancreas sections when you see under the microscope. Okay, so here this pancreatic secretory duct and interlobular duct and large secretory duct. Okay, these are the all duct systems. But what we were discussing is about this acini. So this is a single acini here, and you will find these nucleus are arranged at the periphery, and the central portion is hollow. So all that secretion of these single acinous cells, pancreatic cells are poured in this cap and then they form that tubule system, duct system which are all collected together and they form a major interlobular duct. Interlobular duct means from different lobules, all that secretions come together and unite together and form a single duct, and that duct is lined with the simple cuboidal epithelial layer. But they shows good amount of connective tissue and muscles around that tubule, so we can easily identify that this is a section of a tubule and not of a acini. And when all these such small interlobular duct combined together, they form such kind of a large secretory duct with a simple columnar epithelium. As the size of this duct increases, the epithelial layer goes on increasing from cuboidal to simple columnar. Now, what I was saying about the acinus is this is a acinus, and all these single cells are the pyramidal secretory cells or basic exocrine pancreatic cells. This is a basic exocrine pancreatic cells, and if you see the cell structure here, there is a lot of Golgi vesicle, Golgi apparatus, and endoplasmic reticulums. Then these vacuoles and zymogen granules you will find in a slide of in a section of these exocrine pancreatic cells, and these secretions, zymogen granules, are released on the surface to exocytosis. This kind of release of these zymogen granules, they are termed as a merocrine secretion. There are different types of secretion process happens in a histology. Among that, one is a merocrine. Sometimes entire cell get detached, cell with a secretion get detached and burst. Then sometimes the cell burst and release the secretions. So likewise, there is a different merocrine, endocrine, merocrine secretion here happens with the exocytosis. And now here you can see the arrangement of these acinar cells with the central opening. Now naturally all these openings are guarded by epithelial tissues. So this single unit with around 4 to 5 cells, this is called as a acinus. This is the exocrine organ of a pancreas. Then these acini are supplied with a lot of blood vessels. Capillaries circulate all these acinas. Now in these cells, for identification when you use some particular dyes, That time we can reveal the presence of these basophilic, acidophilic granules in a cells, as well as the zymogen granule. That way we can identify that this is a pancreatic section. Okay, central acinar cells are there. These are the central acinar cells, and these are the peripheral pyramidal cells. Then these are the nerves. One more thing, that in this pancreas are innervated with the nerves also. Okay, then these small tubule intercalated ducts attach with the each acini, and these intercalated ducts collect together and open into the interlobular duct. Now, these intercalated ducts are lined with the squamous epithelium, and the interlobular ducts are they are lined with the cuboidal epithelium. So, in exam, if they ask to draw the exocrine part of a pancreas, you have to draw this diagram. Now, we'll again once revise this diagram to get it understood very well. You have to show the such pyramidal cells, 5 to 6 cells directing centrally with a opening lumen, and you show one of acini. You show this opening here, that passage with a centroacinar cells, and that passage you label as an intercalated duct. Then you combine two intercalated ducts and show here interlobular duct with the different kind of epithelial cell. Here it you show that cuboidal, here it shows squamous. Then for labeling, you label this pyramidal cell as a pyramidal secretory cells. Then in the middle squamous epithelial cell you label as a centroacinar cells. Then these are the interlobular cells, cuboidal cells. Then show that some blood vessels with endothelial cells and RBC in hollow space. Then you show the nerves with the myelinated nerve fibers here. And in pyramidal cells, at the central portion, you show the black color spot that indicates that these are the zymogen granules. Hope you can able to draw this diagram in exam or in your notebook for the journal purpose. Okay, now when you will observe this slide, pancreas slide under microscope, that time how to identify that this is a pancreatic cells, pancreatic tissue. Then see here, all these are the acini. These are all acini. Why these are acini? Because their nucleus are parked at periphery. All nucleus are at the periphery with central lumen opening is there. Now, if you carefully observe somewhere you will find that intercalated duct, that is no acini, but the cells with the cuboidal structures are there. Epithelial cuboidal structures you can see here. Now here it is a blood capillary. Here again the centroacinar cell is there, and they form this intercalary duct, which goes and interlobular duct form, and then it goes a major pancreatic duct and like that. Now all these dark color portion is the accumulation of some granules or secretions here, and these are the acinar cells or pyramidal secretory cells are there. Now here, by using some dyes, you can clearly see the presence or accumulation of zymogen granule. That way it helps us to identify that this is a pancreatic section. And the nucleus, here in a electromicrograph, you will see this zymogen granule accumulation in such black spot with all this endoplasmic reticulum, then Golgi complex and everything in the nucleus. Okay, think this is a repetition, so we'll skip it. Now, production of pancreatic hormone by three cell type. Okay, so we finish with the exocrine part of a pancreas. Now we'll start discussing with the endocrine part of a pancreas. And already I stated that endocrine part in a pancreas is a isolate of Langerhans, where you will find these alpha cells, beta cells, and delta cells. Okay, so like again, alpha cells in a isolate of Langerhans, they are responsible for production of a glycogen, whereas beta cells are responsible for the production of insulin, and delta cell produce somatostatin. Okay, so for reference, you try to remember Alpha cell, endocrine cells in pancreas responsible for glycogen, beta cell responsible for production of insulin, and delta cells production for the somatostatin. Now here is an isolate of Langerhans cross sections in a diagrammatic presentation of isolate of Langerhans. Now around this isolate of Langerhans, here in surrounding you will have that acinus present, which is an exocrine part of a pancreas. And in between the scattered acini, you will find some small part of such specialized structure that is called isolate of Langerhans. Now, in this isolate diagram, if you use a particular dye, you can separate the presence of these three different types of cells. Okay, so A cell, B cell, and D cells. So here the dark color structure. These dark color cells are the delta cells. You will see that they are arranged on a periphery. Then these glycogen producing cells, A cell, alpha cells are also arranged on the periphery, and in central location there are distributed that beta cells which are responsible for the production of insulin. Okay, so three cell types are present: glycogen secreting, B is insulin secreting, and B is somatostatin secreting. A and D cells are located around the periphery of this, and B cells are located in the central portions. Venous return containing insulin flows by the A cells, alpha cells on the its way out of the isolate. Now, this is the arterial blood flow which goes inside and it's sprayed over the isolate of Langerhans, forming the capillary bed, and again they unite and they produce the venous flow. So through this capillary portion, there is a exchange of hormone take place. Now diagrammatically, you can show this isolate of Langerhans like this. Here, these are the acini or acinas spread around the isolate of Langerhans, and you can see here the capillary entering from all the side of acini into this central island. Here in island, you will find these different cells are scattered among the capillary network. These scattered cells are of three different types. We just have seen that beta cells, this blue color, alpha cells, red color, and delta cells somewhere here that is responsible for the production of somatostatin. D cells or delta cell also responsible for the control of secretion of this insulin and glucagon. Okay, now how these alpha cells and beta cells are attached with this capillary. So do you remember the type of capillary here? It is a fenestrated capillary, the one which we have seen in a glomerulus in a kidney. So these beta cells surrounds this fenestrated capillary, and there they exchange their secretions. They pour that insulin in the capillary. They pour the glycogen in this capillary. So these are the alpha cells. This is a vascular pole for secretion by exocytosis. That area which is joined toward the capillary, that is called as a vascular pole. Okay, so the insulin that increase the membrane permeability to glucose. I think this is a physiology point. What is the role of insulin? They increase the absorption of glucose. They promote the glycogenesis and decrease blood glucose level. They withdraw all your sugar present in a blood, and glucagon that promote glycogenolysis. They increase the blood glucose level. These two hormones are antagonistic hormones. If there is a secretion of glycogen, there is a secretion of insulin. If there is a less secretion of insulin, there is a less secretion of glycogen. That is what antagonistic thing is. Now, this cross section, in this cross section, you will see actual slide cross section. You will see here isolate of Langerhans. These all are the acini surrounding. All dark color structures are acini, and in this middle portion, there are scattered cells you can see here with a lot of space in between. This space represent the capillary portions, and all these cells represent the alpha, beta, and delta cells. Okay, here again, less amount of granule and less cytoplasmic coloration is there. That way you can identify this as a isolate of Langerhans. Okay, this is how isolate of Langerhans looks like in a scanning electron microscope. Okay, so this is the beta cells. This is alpha cells. You can identify here alpha cells with a particular dyes, and here you can see these beta cells stained in different color.